Hello, Dr. Mark Kontos with iTube Daily Coverage here at the ACO Symposium in Berlin. And with me today is Dr. Marco Lombardo in practice in Rome. And we're going to talk a little bit about interaction between ultraviolet light and riboflavin in regards to corneal crosslinking. Marco, thanks for joining me. And of course, collagen crosslinking is a very, very big topic right now. And of course, in the United States, we're waiting for approval. Studies are going on. There's a lot going on. We're learning more and more and more, of course, but you guys are much farther down the road. And you've been doing some research looking specifically at riboflavin interaction with ultraviolet light in the cornea and kind of finding out some important information. Tell me a little bit about what you've found.
Yes. Our studies were aimed at understanding some of the specific properties of the cornea in relation to the UVA radiation for corneal crosslinking. From our findings, we improved the understanding of the clinicians and researchers on which will be the factors that have to be taken into account to enhance the outcome of corneal crosslinking. These factors include the meridional anisotropy in UVA absorbance of the human cornea and also the role of the corneal epithelium in filtering out the UVA. Why I choose these two factors? Because we need to take into account the contribution of the epithelium, because now the research is moving to find safer procedure for corneal crosslinking that will require not to remove the epithelium. But the epithelium filters out 20% of total UVA of the cornea. So we need to change the radiation protocol for the transepithelial corneal crosslinking in comparison with the conventional one in which we remove the epithelium. At the same time, a Gaussian beam profile is not adequate to deliver the same amount of energy throughout the corneal tissue, because the absorbance of the cornea increases as the distance increases from the optical center. We found 4 mm from the optical center that the UVA transmittance decreases of about 1/3 with respect to the optical center. A big part of our work that is actually ongoing is what happens when we soak the cornea with riboflavin between tissues with or without intact epithelium. So far, all the researchers and manufacturers are comparing the results of transepithelial corneal crosslinking with those of the conventional one. From our preliminary work, we must change our mind, because the optimal riboflavin concentration is different between the two procedures. Because in tissues without epithelium, we need more riboflavin that also act as a mask, as a filter. So part of the riboflavin will be used to produce riboflavin radicals, reactive oxygen species, to form additional cross-linking bonds between stromal proteins. But a part will be used to mask the underlying endothelium, and so to minimize the phototoxic effect on the endothelial cells. With the epithelium, we have a natural filter, so we don't need the same amount of riboflavin concentration as in the conventional procedure. Our work and some of the results have been already published in some scientific journals is showing that the concentration between the two procedures is different. But at this time it's also important to understand whether our results shown in the lab are also effective in patients. So we actually completed a randomized clinical trial in patients comparing the conventional corneal crosslinking with a new transepithelial corneal crosslinking, because what happens in patients in terms of safety and efficacy over a long time is at the end the most important results for a clinician.
Yeah, well, it's interesting work, and obviously that's, the more we can learn about how cross-linking is working on that level and what we need to do is in terms of riboflavin concentrations and amount of UV light and all those things are important for us to really kind of get that perfect ratio. I appreciate you taking the time to visit with us on that.
Thank you.
Thank you.